fighting stupidity for all eternity. We are the Cell Sisters, we'll help you learn it quicker.
I've been wondering, what are we? Well, we're made of cells.
Oh.
What are cells? Cells are the building blocks of life. Wow, that must mean they're important. How do we make more?
Okay, so there are two main types of cells, prokaryotic and eukaryotic. They each divide in different ways. But in general, cells create cells through the division of the cell cycle. Before we learn about that though, let's go over some basic things to understand. A chromosome is a single strand of DNA. A centromere is a central structure in a chromosome. We count chromosomes by the number of centromeres present. Each DNA strand is made up of a chromatid. Centrosomes are where microtubules are organized, and in animal cells, they also carry centrioles. Microtubules give cells shape, allow them to move, and help in transport. Kinetic core, along with non-kinetic core microtubules, make up the spindle fibers of the spindle apparatus. Now on to the cell cycle. There are two parts to it, interphase and the M phase. Interphase is when your cells are doing their normal functions. It's split into the G1, S, and G2 parts, and it's 90% of the cell cycle. G1 is where the cell develops to differentiate itself. The cells have two sets of chromosomes that are made up of one chromatid each. It goes through a G1 checkpoint, and if it doesn't pass, it moves to G0 and will not continue to divide. That's harsh, but necessary. Once the cell passes G1, it has to divide and enter the S phase. Here, there's no such thing as turning back. The cell replicates its DNA in this phase, so now there are two chromatids for each chromosome. This is DNA synthesis. Then it moves to the G2 where it prepares for division. There's another checkpoint here. If DNA is damaged, the cell will either try and fix itself or destroy itself through apoptosis. It sounds bad, but if cells are allowed to divide with errors in DNA, the entire organism will fail. It is ultimately sacrificed for the greater good.
After interphase, there's the M or mitotic phase. This phase is split into mitosis and cytokinesis. Mitosis is where the nucleus divides, and cytokinesis is where the cytoplasm divides. What's the nucleus? And what's the cytoplasm? The nucleus is where the DNA material is contained. Cytoplasm is where the organelles are stored. It's the fluid that fills up the cell. Here's mitosis in action. What just happened? It looks complicated, doesn't it? Let's go slower and take a deeper look. First comes prophase. In addition to all the things listed, the centrosomes also move to opposite sides and form the mitotic spindle, which attaches to chromosomes at the centromere. Next is the prometaphase. At this point, the microtubules can manipulate the DNA. The most notable part about this phase is the chromosomes moving to the middle of the cell. Then there's metaphase. This is where the centromeres are on the middle of the cell, called the metaphase plate. After that is an M phase checkpoint. The cell checks if the chromosomes are lined up on the metaphase plate and if they are connected to a microtubule. Otherwise, the sister chromatids won't be separated during anaphase and on. It moves on to anaphase where the cell stretches and separates the sister chromatids to make 4 sets of chromosomes. Don't be too excited though. This only lasts until there are 2 nuclei. Finally, there's telophase. And just like that, mitosis is over. Whoa, I've been enlightened. We're not done yet though. There's still cytokinesis. It starts in the late stage of telophase. The quirky thing about cytokinesis is that it's different with plants and animals. Can you guess why? Because plants don't have feelings? Close, but no. It's because plants have stiff cell walls that don't allow them to easily split down the middle and divide. Instead, they combine Golgi vesicles to make a cell plate in between the 2 nuclei that are separated in mitosis, like they are building a wall rather than just pinching in. On the other hand, there's only a cell membrane in animal cells, so they can simply pinch in half and separate. The ring of actin microfilaments allow this to happen. They shrink to pinch off the cytoplasm. This causes a cleavage furrow, which is a depression in the cell's surface. Got it? Yeah.
Wow, best person ever. Wow, best teacher, sensei, amazing, fantastic, spectacular, incredible, truly marvelous.
But wait, isn't mitosis an asexual reproduction? All the baby cells would be identical to the parent, right? That's no fun. Diversity is important. I want to learn about meiosis. And why did we even learn about mitosis in the first place?
Well, they're pretty similar. Meiosis is where organisms produce gametes. Interphase is the same as in mitosis. After interphase, there's meiosis 1 and meiosis 2. Meiosis 1 starts with prophase 1. Homologous chromosomes pair up making tetrads in a process called synapsis. Everything that happens in prophase during mitosis happens in meiosis as well. Let's take a closer look at the tetrad. We have 2 chromatids that overlap at a place called the chiasma. The chromatids cross over here by exchanging DNA. These new chromatids are called recombinant chromatids. This is a super cool step because it's the first place where genetic variation occurs. Next is metaphase 1. Like in mitosis, the tetrads line up in pairs along the metaphase plate. One microtubule is attached to each tetrad. Also make sure you remember that the chromosomes line up in random order. If one tetrad is here, the other tetrads can still go wherever they want to be. This is called law of independent assortment, and it's a source of genetic diversity. Then there's anaphase 1. This is basically the same as the anaphase in mitosis. After it's telophase 1. This is also basically the same telophase in mitosis. Finally, there's cytokinesis 1. Again, this is where the cytoplasm splits in the middle of the cell and the nuclear envelope reforms. So, 2 genetically different haploid daughter cells are created.
But wait, that can't be the end. You said there were 2 parts to meiosis.
Wow, you were listening. You're right, there is another part. It's called meiosis 2. Cells go straight into it after cytokinesis 1 without going through interphase again. During prophase 2, centrosomes start moving towards poles of the cell, and the mitotic spindle starts to form and attaches to chromosomes at the centromere, and the nuclear membrane breaks down. In metaphase 2, sister chromatids line up at the metaphase plate instead of homologous chromosomes, so crossing over does not take place. However, they still line up randomly, so independent assortment happens and creates genetic diversity. Each sister chromatid will be attached to a microtubule, and in anaphase 2, the sister chromatids of each chromosome will be separated and start to move, going to separate ends of the cells. In telophase 2, the nuclear envelope reforms, the spindle goes away, and in cytokinesis 2, the cell splits along their midlines into 4 haploid daughter cells that are genetically different from the parent cells. Yay, we're done! Finally, let's go get some food. I'm hungry. I'm thinking sushi or maybe Krispy Kremes, but honestly... Hey, not so fast. Meiosis produces the sex cells, meaning the egg and sperm cells. They aren't the same, so they're going to be produced differently. We have to talk about oogenesis and spermatogenesis now. They start out the same. The only difference comes with the division of the cytoplasm in oogenesis. Instead of being even, it's uneven. This uneven division happens in both meiosis 1 and meiosis 2. Therefore, you're left with 1 good egg cell and 4 good sperm cells. The 3 bad egg cells are called polar bodies. They basically just chill, so good for them. So why is it uneven? Well, the cytoplasm basically stores everything. Like literally everything. I'm talking protein, RNA, DNA, nutrients, the kitchen sink, your mom, and there has to be enough of that everything in the embryo. So the cytoplasm is split to give more to one cell. Again, it's basically a sacrifice for the greater good. The sperm cell doesn't even help the embryo during the first couple of days. It's on its own, so it really needs that extra cytoplasm. Wow, so strong and independent, like me. And that's everything. Everything? Like all of mitosis and meiosis and oogenesis and spermatogenesis? Yeah, pretty much. So what do you think? I think I've learned so much that my brain is going to go through apoptosis. I love it.